Our guest expert today is Dr. Klaus Wiemer. He is a world-renowned reproductive embryologist. Dr. Wiemer received his PhD from Louisiana State University. He is certified as an HCOD and also as reproductive embryologist by the American College of Embryology. He has over 35 years of experience directing IVF laboratories and has been an off-site director of several fertility clinics. Dr. Wiemer has published over 80 peer-reviewed articles, book chapters in a wide range of topics in clinical embryology. He has received numerous awards for his research, including an overall grand prize awarded by the American Society for Reproductive Medicine, as well as Life Award for Laboratory Innovation in Infertility and Embryology. His main interests are in embryo culture systems, improving laboratory outcomes, and studying the effects of follicular stimulation on subsequent oocyte and embryo quality. He is currently conducting research on developing an embryo morphology grading system that can be used to predict ploidy, as well as rate and quality of blastocyst development. He was a founding partner of Northwest Center for Reproductive Science, as well as POMA Fertility. He now consults various IVF centers nationally and internationally, as well as related industries. He is a frequent speaker at national and international meetings. Klaus, thank you very much for accepting my invitation to this interview.
Dimitri, thank you so much for having me. I'm super excited to be here and get to talk to our American College of Embryology colleagues. It's going to be great. Thanks.
Thank you, Klaus. Well, with all the contributions you made to this field, for me personally, one stands out as probably the most consequential, which I believe will be your legacy in the field. And this is your culture medium. And my first question will be, please set a stage for us. Tell us why and when you decided to tackle culture medium.
So when we got started, and this is a great question, because when I got started in IVF, I was still a graduate student at Louisiana State University, and I had gotten a phone call from Jacques Cohen, who had at the time had just moved from Bourne Hall to Atlanta, Georgia. He was working at Reproductive Biology Associates. He had read some of my work, and he reached out to me and he says, do you want to come to Atlanta and do some, embryo work with us, some embryo culture work? And I said, yeah, because at the time I had been developing a co-culture system. So my approach initially into embryo culture was not necessarily to try to improve a certain culture media, but was to see that if I used oviductal or uterine cells, if that culture, if the interaction of the media as well as the cells could improve upon the development of embryos using the standard medias, because I wanted to try to understand what exactly was missing or not missing in embryo development. And the reason why I mention that is we found, for example, back in those days, if you used Ham's F10, which is a very complex media for cell culture, if you added oviductal or uterine cells in virtually any animal model, the rate of embryo development was huge. So we were wondering, either the cells are putting something into the media, they're taking something out of the media, or it's a combination of the two. Or retrospectively, we thought about it and thought maybe it's reducing the oxygen tension, around the embryo because of the cell activity. So the first approach was really to see could we take existing culture medias and try to improve on that? Because at the time you had two schools of thoughts. You were taking really complex medias that were made for tissue culture and trying to modify those for human growth, or you were taking things like Earl's balanced salt solution, which is or media like that, and we found that basically it was a salt solution that had some pyruvate and things like that. And we noticed that when we added monolayers of cells, we were getting development. So the original approach was to see if we could improve the the formulas that were out there at first.
What gave you the confidence that you're the man for the job, that you have the expertise to constructively change the culture medium uh, and improve it?
Well, you know, it's not that I had the confidence. I think it was more the ignorance, really. Like there's amazing people that have dedicated their lives to culture media development. Henry Lees, Whitten, of course, Dr. Biggers, and of course, a friend of all of ours is Dr. David Gardner, and all of them kind of came from within the approach. So I was on the outside looking in, and my advisor said, this is a great way to get into the media because you have no preconceived conceptions of how it should or should not go. You can go into it because sometimes ignorance helps you out. It doesn't predispose you to certain ideologies or certain things. So, as I started to work on medias and started to look at it, I realized that the person that I thought had done the most work that was most beneficial was Dr. Biggers. I felt like the way John had gone around looking at amino acid compositions, how all of these amino acids interrelated with each other, really went very well along with the physiology that the oviduct and the uterus were providing, in that these structures were creating a lot of substrate that were very similar to what is found in culture media. And so at the same time, LSU was part of some multi-center grants where the basis of KSOM media was starting to be developed. And so we were having access to that kind of information, seeing that, you know, these salt solutions in in with, along with glucose, pyruvate, and all of the amino acids would work very, very well. And at the same time, we were studying oviductal secretions in animals when I was at LSU. And we noticed a huge amount of glucose, pyruvate, lots of growth factors, but we found all the amino acids. And it didn't matter where that animal was in her reproductive cycle. And unbeknownst to me, David was kind of working on this on his own on the other side of the world. But again, because I'm ignorant to all of that, because I'm looking at it from my point of view, I had no idea that we're kind of working on things at the same time.
How did you decide that it has to be one stage medium? Because this was going against conventional logic. We knew that fallopian tubes and then oviduct and then uterus, they all have their unique, slightly different concentrations of the pyruvate and the glucose and everything, and different pH. This is why the multi-stage medium at some point became so popular, because it was logical that for each stage you need a separate conditions, and then you come out with a single stage medium. How did this happen?
Well, it came from the fact that we didn't, when we were collecting oviducts and uterus from animals, we were not seeing these huge differences in secretory values. We saw rather that all of these materials were present at the same time. At the same time, having access to, in our discussions, particularly I remember talking to Dr. Godke. Dr. Godke always felt like, you know, first of all, we have to stop comparing embryo development in the oviduct with embryo development in microdrop, because
Mhm.
embryos in the oviduct are not covered with fluid. They're covered in a very thick mucus, and the cilia are moving them along. And and then there's going to be receptor sites on the apical aspects of these membranes. And Andy Watson when he was doing a postdoc with Gil Schultz up in Canada, also confirmed that on the apical aspects of all these bovine blastomeres were receptor sites for virtually everything, and that these receptor sites didn't change in the bovine embryo, either pre-embryonic activation or post-embryonic activation. So again, out of ignorance in my mind, I never even thought that embryos would have a stage 1 and a stage 2, because again, the background of what Andy Watson had found, again, the information that we were learning as Dr. Biggers was working more and more upon the concept of availability of medias. And then adding to that, Dr. Godke kind of saying, stop trying to compare the oviduct to a microdrop because they're not the same. In a microdrop, the embryo is covered, is surrounded by fluid. There's no interaction between the embryos, no active communication, if there is such a thing, but rather that he was convinced that we need to expose the embryo to all the solutions in the right balance, and the embryo is going to pick and choose what it's going to need, because that's what happens in vitro, not in vivo. And so that's what kind of set me on the mindset that I bet you we could get by with just one media.
But F10 medium, if we think about it, this one you mentioned, it seemed to have everything, uh, even like more components possibly than in embryo culture medium, and it did not work for human embryos uh, as it was. So this is why I feel like must be something else which was in play to say that embryo will pick from the culture medium whatever it needs, because I mean F10 would otherwise be uh, suitable for multi-days culture.
Well, to add to that, so at that time, I started to do some co-culture work early on with Dr. Bill Schoolcraft and Terry Schlenker. And Terry reminded me that the HTF solutions that they were using were a modified HTF, that they did not contain certain products like, I think one of them is hypoxanthine.
Mm.
And so then, and so then that got me to go back and exactly like what you just said, too much of something can be bad. So when we started to look at the secretory factors that we found in oviducts and uteruses and compared that to the HAMS F10 and HAMS F12 solutions, we quickly saw that there were a lot of products that were in these tissue culture media that were just simply not necessary and could potentially be toxic for embryos.
Mm.
And so those were quickly removed because we didn't feel that they added anything and that they could potentially be toxic to the whole process, that they could interfere in cellular aspects.
Okay, now it makes more sense. Now I'm getting somewhere in understanding
Yeah.
how it was evolving. And how long could you culture embryos at the time, human embryos, at the time when you started developing the medium, global
Well,
medium?
when we first started, we were a little bit, and Don Rieger was very much involved with this. So, Don also really reinforced the whole idea of, trying to compare the oviduct to a microdrop was kind of, as he said, how Don is, he almost called it
Mm.
nonsensical. And he's right. So what we felt was like, okay, let's take baby steps. So at the time, we had developed a modified HTF media that really worked great for the first 3 days. We were a little bit worried that at first exposing these human embryos to all of these amino acids could potentially be bad. So when we first started in my lab, we started culturing first in HTF, a modified HTF, and then we were moving to day 3 to global meat to global as we know it today. However, Catherine Rakowski, about that maybe just a little bit afterwards, under the direction of John Biggers, said, no, let's take a different route. What we should do now is culture. Let's start in global. Let's refresh on day 3, and then let's, move over. And part of the refreshing was because we didn't have good air handling systems at the time, so we were worried about embryo toxicity, building up in the oil. And B, we didn't really understand how much metabolites were going to accumulate in the microdrops. And we were worried that the embryo would, use up all of its product and basically, could potentially have reduced viability because of all the contaminants, byproducts that are present. Now we know that all these products actually serve a paracrine purpose and actually get these embryos to grow. So when we first started with really simple salts like a modified HTF, and then moved to global, or in Catherine's case, she went directly for it and just changed it till day 3 and moved it on day 3. We didn't really move to single step all the time until we did some prospective randomized trials in some clinics where we had really good air handling systems, and we modified that because again, that's when all the VOC stuff was starting to come out from Antonia Gilligan and Jacques Cohen, and how the oil could potentially be a big sink. We had noticed a lot of effects of seasonality in some of the labs we were working in. So we thought we better fix the air component before we start doing extended culture, because I think that's really important to understand those two together.
Hmm. Interesting. Um, Now, looking at the current status of this culture medium, global culture medium, do you feel that its quality is, or its ability to support embryo development is equal to what is achieved in vivo?
You know, that's an amazing question. And if I can just, if it's okay to just give my own opinion on that, I think the part that's still missing about embryo development in vitro is the paracrine aspect. I'm a big believer in the paracrine route. I'm a big believer in how all these intermediate growth factors that are secreted by the oviduct and uterus really help embryos. And I also believe that the culture systems that we have today impact the lipid profiles a little bit, because the reason why I say that is back in the bovine days, we took some in vivo produced blastocysts and in vitro produced blastocysts. Of course, don't do this with humans, kids at home. What we did is we put them in a dye called trypan blue, and then we dropped them at the top of a graduated cylinder. And the in vivo embryos sink much faster than the in vitro embryos. They always do. And so that
Hmm.
alone made me realize in vivo embryos are heavier. So Yves Menizo, an amazing scientist, contributed a lot to our field, the B2 media. He did some great co-culture work with Vera cells. Dr. Menezes really felt strongly that in vitro produced embryos don't have anywhere near the lipid contents that in vivo produced ones do, and these in vitro embryos, are probably lighter, and so I don't think they're the same. So I really feel like the part that's missing right now are these intermediate factors that are involved, because again, Andy Watson has some great work from the late 80s and 90s showing all these growth factor receptor sites all over bovine embryos. And as we all know, Mother Nature doesn't put these receptor sites there for no reason at all. You know, what we haven't done is done radio labeling studies, say where we tag, say, some kind of epidermal growth factor with a non-toxic dye, and see if these growth factors are incorporated by the embryos, and how does that growth factor move around. Now, adding growth factors to culture media, I'm not telling anybody to do that, because growth factors have, or really, we don't need, like, what's the half-life of a growth factor in vivo, and in what if you add that in media? Are you going to have early or premature activation of certain genes in these embryos, things like that. So I, I don't ever think we're going to get quite to what in vivo is, simply because we can't replicate that paracrine aspect.
Hmm. I mean, I think that it's very important to keep in mind that we're not perfect in culture medium and maybe we'll never able to fully replicate this in vivo development, which makes me ask you another question. Do you think that this additional or even one-day culture, any type of in vitro culture, will select against some traits which otherwise compatible with in vivo development? And some embryos would benefit from being transferred into the uterus immediately after a check of fertilization, or maybe before day 3, as opposed even if they can be left in culture and deal to the blastocyst. In fact, I have it's like two questions. One, do we lose some of the embryos because of in vivo culture? And then second one, obviously, is whether there is a benefit for certain specific traits which embryo may metabolically exhibit for being transferred earlier.
Let me answer the second question first. I'm not really sure about transferring embryos earlier. In fact, because I come from an animal background, if you, for example, transferred an 8-cell embryo, which is normally found in an oviduct, into a cow, sheep, goat, pig, horse, you might as well just throw the embryo on the ground. You're going to have terrible, terrible outcomes. You won't get any pregnancies. So obviously the uterine environment in a lot of species is extremely hostile because I don't think the right factors are present. However, in the human, we've done very well. In fact, Michael Tucker's even talked about years ago about doing pronuclear transfers, in patients. Or I remember when I was living in Florida and we had hurricane, was it was it it was a really big one there. We ended up transferring some zygotes in patients, uh, and we got some patients pregnant, which was beyond me. So I think in the human, there, we've got to remember that the embryo is going to become a blastocyst or not. I I think in certain patients, doing a day 3 transfer or a day 4 transfer might make sense, because they are going to get exposed to some of these, these paracrine routes. And I only say this anecdotally because we all have patients that have failed multiple blastocyst transfers. They don't make blastocysts, or their embryos arrest at the morula stage. And so then we went ahead and did a day 3 transfer on them, thinking perhaps the paracrine route on these embryos is super important. Maybe there's something missing on the glycolytic side or some other metabolic pathway that we don't quite have a grip on that's impacting the paracrine aspect. So we transferred on day 3, early on day 3, so that that embryo would get exposed to whatever paracrine aspects were in the uterus. And we got some patients pregnant. Now somebody's going to argue, well, the embryo had to become a blastocyst in order to implant. My suggestion would be, because I can't even argue, the point is, well, maybe the uterus provided the conditions to do that transfer on day 5 or day 3 that otherwise we couldn't replicate in the lab. So I do think there are a few patients like that.
Okay, I think it's a very important observation, and even if you validate it just anecdotally, it's still important because oftentimes when patient doesn't have embryo development, as you expect to day 5, the decision has to be made like, what is next? Maybe.
And I can
Mhm.
I can tell you this, in abnormal embryos, back years ago when it was a little easier to do research on embryos, we had a IRB approval to work with embryos that patients wanted to discard, or, just didn't want anymore. And, back in the day with a lot of day 3 embryos that were frozen. So, we thawed a lot of these day 3 embryos, and we found that, when you cultured them with growth factors, that they made amazing blastocysts, and many of them started to cavitate late on day 4, that you would have embryos that were clearly ahead of the game. It's just that these embryos that were exposed to growth factors, you would never do this with, with normally fertilized embryos, because we were worried about, what else were we activating? What else were we turning on in these embryos? Because these growth factors are so strong. I mean, not necessarily strong is a wrong word, but very powerful. And we don't know half-lives. And we also don't know what happens if an embryo is exposed to a growth factor prematurely. What does that do downstream in terms of gene activation and other aspects?
Yeah, I agree. I think there are many details which we will be missing forever. We'll never know
Right.
the 100% of everything, but we just have to keep this in mind. And we don't know if it's consequential, which brings me to another question. Do you believe that some embryos would do better in one medium and others in another?
Well, the first part is absolutely right. When we were with global media, we had the potential, we worked with everybody's media, all the competition. And I know they worked with our media as well. And then, when we were doing a lot of trials with labs, especially in Latin America, it was funny how there would always be really beautiful embryos in every type of media. And I remember, some of my colleagues thinking, Klaus, if you had the ultimate way, if you had somebody with 20 embryos, wouldn't it be great if we could culture them in the best 3 culture media? Because I bet certain embryos would have preferences to go down certain metabolic pathways than others. So I'm totally convinced that certain embryos like certain medias, which again answers a question that we haven't completely optimized our medias yet, because I found the same thing. I had certain patients that did much better in a 2-step media than they do in a single-step media. I had patients that do better at a certain pH than another pH. Some patients that do better with uh, pure HSA versus more of the the less defined proteins that contain, the alpha and beta immunoglobulins. You just, if you could have every combination, every embryo would kind of pick its own way um, from that perspective. Uh, can you remind me what the other question was, though?
The other question was whether you observed the little bit different morphology of the
Oh, yes,
blastocyst.
yes,
Yes, depending on medium.
absolutely. So one of the first things that people said to us when they were working with Global was like, "Oh my God, I looked at my embryos on day 3 and the embryo's a blob." And I'm like, "That's what it's supposed to look like. That's called compaction." And they go, "Well, why are my embryos compacting in global media and they're not in my other media?" And that's because they're being exposed to all the amino acids. So the presence of all the amino acids, and this is an idea that Don Rieger had come up with. Don theorized that the presence of all the amino acids was accelerating or keeping more true to what happens in vivo, the development of tight and gap junctions. And because when you look at sectional slices of in vivo produced 8-cell embryos, one of the biggest morphological attribute that you see is the plethora of tight junctions and gap junctions that are forming, which is obviously really involved in that next step of development. So I absolutely think that the components of a culture media will totally change how an embryo appears. I see the same thing with the inner cell masses. For example, in global media, we tend to see sometimes these patchy, large trophectoderm cells that almost look like a squamous cell. If you culture that embryo for a few hours, they go away. Whereas other kinds of media, you don't necessarily see that. You see that classic epithelial shaped trophectoderm cell. So 100% behind you. I think the embryo is a product of the constituents that are present in the ratio that they're present. And then also how that interacts with how clean the proteins are.
This observation, what you said, makes me wonder if there is a preference of an embryo for a mother. So maybe some embryos just just happen to be in the wrong mother, and which doesn't have the right combination of nutrients for this embryo to do well. And maybe in these patients, they do need in vitro culture, which will compensate for that.
Yeah, it's funny you say that because when I was, back in the 90s, we had some patients that had done multiple cycles with their own eggs and they had reached a point where they couldn't afford to do IVF anymore. And so they said, is there any way that I could be a donor for a patient? Now, again, this is back in the days when, obviously you wouldn't have used a multiple failure patient as an egg donor, but out of desperateness, we did a couple of patients like that. And what was really sad was that a lady who had used her own eggs 3 to 4 times to try to get pregnant never got pregnant with her own eggs. But yet, if she donated half of her eggs to another couple, the couple walks away with a baby. Now, maybe it was a uterus, maybe it was a sperm, but it was kind of like, what the hell? How does that happen? What again though, and it's something we haven't really talked about. It makes me think a lot like how important, because we talk so much about the egg and stuff, but we don't really talk about the contribution of the sperm, so much to the egg and to the embryo itself. But, it makes me wonder, like, these patients that don't get pregnant, despite the fact that they make beautiful embryos, if there's some kind of etiology with the sperm or the uterus that, we just can't figure out, because we know with horses, for example, recipient mares that have high amounts of lactobacillus and other beneficial bacteria in the uterus have much higher conception rates than horses that don't have that. But yet in the human, we haven't really been able to show like the ERA or microflora or any of these things that would really pick out like this is an optimal uterus, this is not.
How is your animal experience help you to succeed in human IVF?
Oh man, that's a wonderful question. The animal experience was really good because it taught me if you're an animal scientist out there and you've done this, you know exactly where I'm coming from. You have to learn how to manage your animals, you have to learn how to manage your recipients, and you have to manage all that with your laboratory. So what it does as a laboratorian, it really makes you realize that you've got to coordinate the treatment with your laboratory. Whereas if you don't have animal experience and you go straight into an IVF lab, you totally lose the appreciation of all the work that the nursing coordinators do and the rest of the clinical staff really do to ensure that when that patient shows up for her egg retrieval or embryo transfer, that everything is optimized. Whereas if you're an animal scientist, you're one day out there, you're a herd health manager, and then you take your coveralls off and you run into the lab and now you're managing embryos and you've got to make sure that you've got recipients that are lined up for this stage of embryo or this or that or the other. And these are conceptual things that makes you pull in your treatment plans with your donors and your recipients, which the earlier as an embryologist you get that, the more appreciation you're going to have on understanding synchrony of the uterus for frozen embryo transfers, or understanding, how patients are organized and stuff. So I think it really helps a lot from that perspective. And then working with animals, if you can grow bovine embryos and bovine blastocysts and understand all that, it really helps you work out aspects of sperm preparation, embryo culture. You get to troubleshoot a lot. You can make some really bad mistakes and not have too big of consequences of it. I think it's just this amazing training for the real world in all the ways I just described.
And what about your expertise in ovarian stimulation? Did it start with animals?
If you're an animal scientist doing this kind of work, you do the follicular stimulations, you do the egg retrievals, you grow the embryos out, and you also do the embryo transfer. So the thing that I loved the most about this was that because I had to do the follicular stimulations, I got very involved in that because that was super important. In other words, I wanted to get my PhD, so I needed to figure out like how to get 30 embryos from a cow, not 5. So by learning that management tool, I learned a lot about physiology and how it works. I was really blessed early on in my career, though, when I was working with Jacques Cohen at Cornell University, that Dr. Rosenwax, who I will owe this to forever, he took me under his wing because I showed a lot of interest in follicular stimulation. And Dr. Rosenwax allowed me to sit in with him, during when he was teaching the fellows. And, he's like a guru on this. And it turns out that the human and the bovine have very similar aspects in terms of follicular recruitment and things like that. Cattle have a little FSH peak during the mid-luteal phase, just like humans do, which we think is used to recruit the next crop of follicles. And so in the bovine, we're constantly manipulating that window. And those were things too that, Dr. Rosenwax was trying to teach the fellows, like how to maximize that window, how to take advantage of it, when not to start a patient. He actually was like one of the first people I said, this is a bad cycle. She's not going to do well on this. And so, I think it started with the animals, but it really was finely tuned by Dr. Rosenwax and then later on by Dr. Bill Schoolcraft. They both were super awesome in teaching me a lot about human follicular stimulation.
All right, so it's your personal animal experience with ovarian stimulation, plus then layered by experts in human stimulation. So you have this very unique vision on the process. So
Yeah,
that's
and
mhm.
and to all the young biologists out there, I think it's super important that you understand follicular stimulation. You need to understand that, why is it that you can get 30 eggs from a donor and what's the consequences of that? How does that happen? Are those eggs the same? Are they different? Maybe that's the reason why some, embryos that are genetically normal lead to a miscarriage. But again, if you don't understand the basic physiology behind follicular stimulation in your laboratory, if you're a lab director or lab supervisor, it makes it very difficult for you to troubleshoot and to understand why are things going well and why are things going badly in the lab. And if you don't understand that
Definitely.
science, you're never really going to understand how your lab works, in my opinion.
Well, absolutely, I agree with that. Embryologists are in a unique position to be able to interpret what they see in laboratory in context of the patient history, including ovarian stimulation. Reproductive embryologist, somebody who can actually do that. Last question is, and this is a little bit outside of the focus of this presentation, but I'm sure it's interesting for a lot of people. Do you believe that it is possible to completely automate IVF laboratory?
I think it's possible to automate the laboratory in stages. So in other words, so I know I'm not answering the question completely. So I think very, very quickly, we're going to see the automation of things like dish preparation, ICSI, things like egg retrievals. I have no problem with that. I think that's going to happen quickly. It's so the long answer for this is I don't really know. And the reason why I say I don't know is more ignorance on my part. In theory, if you have enough robots and AI to train, I think you could pretty much teach a robot to do anything, automate anything such as, biopsies, tubing cells, things like that. but I think we're such a long ways from that that I think in our lifetime, what we'll see happen pretty quickly will be a partial automation of the IVF lab with the goal to go fully automated. But I think to go fully automated, and I'm probably going out on a limb to say this, will require skill sets that still have not been developed yet. You know, the complexity of skills. You know, when you're biopsying an embryo, you're thinking about three dimensionals, and you're constantly thinking 2 to 3 steps ahead of what you're doing. And you know, I just don't know enough about the AI aspect of that. Like, how would you teach a robotic structure to anticipate that this cell is about to break loose, or that little piece of trophectoderm is about to come loose? Um, I think we still need some lacking, but at some point, I truly do think that IVF labs will be truly automated. That they have to become fully automated, or else we're never going to meet the demand.
Okay. Okay. Well, Klaus, thank you very much for this interview. It was a pleasure to see you, to be able to ask you questions. Uh, Thank you so much.
Dimitri, it's been a pleasure. Thank you for giving me the opportunity to go down memory lane and think about a lot of stuff. And then, you know, to all the young embryologists that are out there, you know, please reach out if you ever have any questions. I'm here to help any way I can. I'm at the point in life where I want to give back. I stood on the backs and shoulders of many giants to get to where I am today. And so I just want to tell everybody I got a back for you guys to stand on, because we need some more giants. We're really needing some giants out there.
Well,
Thank you so much,
you are
Dimitri.
definitely the one. Klaus, thank you so much. Bye-bye.
Thanks, pal. Bye bye.